Okay, let's jump into this deep dive. Today we're exploring something really at the intersection of cutting-edge tech and, well, essential medicine. We're looking at how artificial intelligence is being used to analyze blood samples, specifically those detailed peripheral blood films, PBFs.
Yeah, it's a fascinating area. It's really seeing a surge in research lately. So we've gathered a collection of recent studies, literature reviews, to kind of get a handle on the current picture.
Exactly. So our mission here for this deep dive is really to go through these sources with you, pull out the core insights. We want to get a sense of the huge potential AI has here, what it's already doing, but also, and this is key, the big challenges it still faces. Before it becomes just a standard tool in the lab,
hmm
think of it as us distilling the latest knowledge on AI and blood cells straight from the research.
Right. And maybe to start, we should ground ourselves in why looking at these blood films is so important anyway. You see, a peripheral blood smear examination, it's just fundamental,
Yeah.
absolutely fundamental for evaluating blood cells.
Okay.
It's that critical step after the automated complete blood count, where machines give you the raw numbers. This is where you actually look at the cells.
Right, so the machine gives you counts, like red cells, white cells, platelets.
Yeah.
But then a human looks at the smear under a microscope to see, well, what they actually look like. The shapes, the details.
Precisely. That visual inspection, the morphology, it's crucial for confirming those machine findings, spotting abnormalities the counters might miss, and of course diagnosing a whole range of conditions, things like infections, different blood disorders, even cancers.
Okay, so it's clearly vital.
But doing it manually? Well, even with expert hematologists doing the really detailed morphology work, it's tough.
What makes it so hard? I mean, beyond just needing that expert eye.
Well, first off, it's incredibly time-consuming. You're scanning a slide, visually IDing, counting different cells, looking for subtle, often rare abnormalities.
Right.
It demands huge expertise.
Mm.
And it's also, prone to human error. Think about observer fatigue, long shifts.
Yeah, I can imagine staring down a microscope all day.
Exactly. And then there's access.
Yeah.
Getting that level of expert opinion, especially if you're in a remote area or an underserved region, that can be a major, major bottleneck.
So let's say a sample flags something on the automated count and they decide they need that manual smear check. What's the sort of typical delay we're talking about?
Just for that manual confirmatory step, it can easily add around, say, 3 to 4 hours to the whole process.
Wow, 3 to 4 hours just for the smear review.
hmm
Which I guess is exactly where AI starts to look really attractive, right? Offering a way to potentially automate this, get around those manual hurdles.
That's the core idea, absolutely. Applying AI, machine learning, to analyze digitized PBF images.
Mm.
And what's really interesting digging into these sources is that this isn't like a brand new idea. The very first AI study looking at PBFs, it actually dates all the way back to 1972.
1972.
Oh,
Seriously,
cool.
wow, that's way earlier than I would have guessed. What were they even doing back then with AI and blood cells?
Well, that early work was focused on trying to automate leukocyte differential counts. Basically, getting computers to classify the different types of white blood cells from images.
Okay.
Obviously, the tech, the capabilities, they've grown exponentially since the 70s, especially with deep learning coming along, things like convolutional neural networks, CNNs. They're just incredibly good at image analysis.
So the modern approach is what? Take a really high-resolution picture of the blood smear and then train an AI model to essentially see and understand what's in that picture, like a digital pathologist.
Yeah, pretty much. Using computer vision, deep learning techniques to classify cells, detect them, count them, all directly from those digital images. And the aim is to replicate, or maybe even, exceed human performance in some ways, potentially much, much faster. And looking across the research we reviewed, AI is being applied to a really broad range of tasks in PBF analysis. The rapid review paper, for instance, it covered studies on malaria detection, different leukemias, various white blood cell types, leukocytes, red blood cell shapes, erythrocytes, even things like myelodysplastic syndrome, or MDS, and studies looking at mixed cell populations too.
Okay, and this is where it gets really interesting, right? The performance numbers. Because one source summarizing studies reported some pretty impressive self-reported mean accuracy rates, like really high.
They are quite striking in research settings.
Yeah, we're talking averages around, what, 95.5% for malaria detection, 96.0% for leukemia studies,
Mhm.
0.94% for general leukocytes, 95.2% for mixed cell studies. Even identifying different red blood cell shapes, that was hitting 91.2%.
And the overall average across all those different tasks came out at 95.1%.
That's pretty remarkable for such complex visual tasks.
It is. And the sources really highlight some specific wins. AI has shown really high accuracy in detecting and classifying different leukemia types. Acute lymphoblastic leukemia, ALL, comes up a lot as a strong point.
Okay.
It's also doing very well classifying multiple subclasses of leukocytes. Neutrophils, eosinophils, basophils, lymphocytes, monocytes, the whole gang.
And what about picking out those, funny-shaped red blood cells, the ones that indicate specific problems?
Yes, identifying various abnormal erythrocyte morphologies, things like sickle cells, elliptocytes, acanthocytes, those spiky ones. Some studies, usually the ones focusing on a very narrow specific classification task, they even report hitting 100% accuracy, though that's often under very controlled conditions.
Still impressive, and just like finding and counting cells, platelets, for example.
Yeah, AI shows real potential there too. Studies have definitely achieved high accuracy in detecting and counting specific cell types. One source even highlighted a study reporting 99.9% accuracy just for platelet segmentation.
Segmentation, what's that exactly?
It's basically the AI drawing a precise boundary around each individual platelet in the image. So incredibly accurate identification of where each one is.
Okay, so we see what AI can do handling all these different cells, hitting these high accuracy numbers in studies. It makes you wonder, how exactly is it doing it? What are the techniques sort of under the hood?
Well, the sources make it pretty clear there's a dominance of deep learning techniques,
Right.
especially, as I mentioned, convolutional neural networks, the CNNs. These are the kind of networks that has basically revolutionized image recognition everywhere, not just in medicine.
Right, like the tech that recognizes faces in your photos or identifies objects, but trained specifically on blood cells.
Exactly that principle. And the typical workflow, the process, usually involves a few key steps. First, there's image preprocessing. That's about standardizing things, maybe resizing images, cleaning up noise, enhancing the contrast, just making sure the images are consistent so the AI can learn effectively.
Makes sense. You need clean data to start with. Garbage in, garbage out, as they say.
Precisely. Then, a really crucial step, particularly in medical imaging, where you might have rare conditions with limited examples,
Yeah.
is data augmentation. This is where you take the images you do have and you digitally create variations. You might flip them, rotate them slightly, zoom in or out a bit, maybe tweak the brightness or the color saturation. One source mentioned mirroring hue saturation exposure changes specifically.
Ah, okay. So you're essentially showing the AI the same cell, but under slightly different conditions to make it more robust.
Exactly. So it learns to recognize the key features of, say, a blast cell, no matter if the slide's a bit differently stained or lit or the cell's rotated.
Clever. What comes after that?
Often there's segmentation. That's about isolating the individual cells or regions of interest within the bigger image, like drawing those boxes around the platelets we mentioned.
Got it.
Then the AI performs feature extraction. This is where it learns what visual characteristics actually define a specific cell type, its size, its shape, the texture of the nucleus inside, the color of the cytoplasm around it, all those subtle details.
Okay.
And finally, using those learned features, it performs classification. Assigning the cell it just segmented and analyzed to a specific category, neutrophil, lymphocyte, blast cell, whatever it might be.
Were any specific algorithms mentioned often, like particular types of CNNs or other methods?
One algorithm that popped up was YOLO, stands for You Only Look Once.
You only look once. Okay.
Yeah, it's known for being a really efficient, real-time object detection algorithm. It can look at the whole image at once and identify objects and their locations. And one source specifically highlighted its reported robustness, saying it performed well even with lower quality or slightly blurred images, which, is a big plus for dealing with real-world lab variability.
Okay, so the techniques are clearly powerful, leading to those impressive accuracy figures we talked about. But like any AI, its intelligence really depends on the data it learns from, right? And I get the sense this field has some pretty unique data challenges.
Oh, absolutely. Data is foundational. And frankly, it's one of the major bottlenecks right now. These studies, they rely on having large, well-annotated image data sets. Basically, lots of pictures of cells that experts have already labeled correctly.
Are there public data sets people use?
Yes, some common public ones get mentioned, like LLIDB, which focuses on leukemia, and CNMC. These are really valuable resources for researchers.
But I sense a but coming. The sources point out issues with the data that's currently available.
Significant issues, yeah. A really key challenge highlighted across the board is that many studies rely on data collected from just a single medical center.
Right.
So you might train an AI model using thousands of images from Lab A, and it gets incredibly accurate for Lab A's specific way of preparing slides, their staining protocols, their scanner.
But why not work as well if you try to use it in Lab B down the road?
Exactly. It might struggle with samples prepared slightly differently, using different equipment, or even from a different patient demographic.
Hmm.
So generalizability, whether the model works reliably elsewhere, is a huge question mark.
Okay, so lack of multi-center data is one thing. What else?
There's also just a shortage of high-quality, expertly annotated data for the rarer stuff. Rarer cell types, less common morphological abnormalities, non-malarial blood parasites. If the AI hasn't seen enough good examples of these things, clearly labeled by experts, it simply can't learn to identify them reliably. This really limits its potential diagnostic scope beyond the common findings.
And I think one source mentioned private data sets being an issue too.
Yes, a lot of valuable data sets are developed in-house and kept private, which is understandable, but it makes it really hard for other researchers to independently verify the performance of models trained on that data. It hinders reproducibility and building trust.
Are people trying to fix this? Like creating new, better data sets?
Definitely. Efforts are underway. One source described the development of a new, large annotated data set they called BioNet. It was specifically created to provide more high-resolution annotated images, focusing on things like cell detection and counting, aiming to help fill some of these data gaps.
And didn't the Leukemia Review find something really telling about using multiple data sets?
It did, yeah. That was a fascinating point. The review found that studies which trained their AI using multiple different data sets tended to achieve superior accuracy. Their average accuracy was reported at 96.72% compared to 94.98% for studies that only used a single data set.
Wow, that's a noticeable difference.
It really is. And it strongly suggests that feeding the AI more diverse data from different sources is absolutely key to building models that are more robust and more likely to generalize well to new, unseen data. So while those overall accuracy numbers, like the 95%, sound great, for actual clinical use, you really have to dig deeper than just that single number. What becomes critical are performance metrics like sensitivity and specificity.
Right, sensitivity being how well it catches the true positives, the actual disease or abnormality, and specificity being how well it identifies the true negatives, avoiding false alarms. Why are these so crucial in medicine?
Well, think about it. Low sensitivity missing a positive means you could miss a diagnosis. A patient with leukemia might get missed. That's obviously a huge problem.
Absolutely critical.
But low specificity, too many false positives, is also a big issue. It means lots of unnecessary follow-up tests, huge patient anxiety, and it could totally overwhelm lab staff chasing down flags that aren't real.
Okay, so you need both high sensitivity and high specificity for it to be actually useful in a clinical setting.
Precisely. You need that balance.
So how did the AI systems stack up on sensitivity and specificity based on the sources? Did they give examples?
Yes, one source that evaluated a specific decision support system gave some concrete numbers for certain cell types, which gives us a better feel for it. For some key abnormal cells, the numbers looked quite strong. For myeloblasts, which are important for leukemia, they reported 98% sensitivity and 98% specificity.
That's very high for both.
It is. Basophils were 94% sensitive, 99% specific. Normal blasts, immature red cells, 93% sensitive, 99% specific.
Okay, so really promising for those specific cell types.
It does seem that way,
Hmm.
but there was more variability for others. Lymphoblasts, another leukemia indicator, showed 90% sensitivity and 92% specificity in that system. Still good, but not quite as high.
Right.
And for specific abnormal red cell shapes, like target cells, it was 81% sensitive, but only 79% sensitive, but it had very high, 99% specificity.
So it shows the AI can identify these things, but the performance isn't uniform across the board. It really varies depending on the specific cell type.
Exactly. And the sources also pointed out that even the commercial AI analyzers, the ones that are already approved as medical devices, while they show high accuracy for common cells, they still have known limitations with less common cell types or parasites, which again circles right back to those gaps in the available training data for the rare findings.
Okay. So we've painted a picture of immense potential, some genuinely impressive results in research settings, but you've also hinted that this tech isn't just plugging into every lab tomorrow. This feels like the reality check part of the dive. What are the major roadblocks stopping wider adoption right now?
Yeah, this is where the challenges really come into focus, and the sources are pretty consistent on the main hurdles. First and foremost, the data issues we've touched on. That lack of well-validated multi-center data is probably the single biggest barrier to ensuring these models are truly generalizable and reliable everywhere. Then there's the lack of standardization. Different labs use different scanners, different image formats, different data protocols. It makes it incredibly hard to build AI models that work consistently across sites, or even just to compare results from different studies fairly.
And the private dataset issue hinders verification.
Exactly. If you can't test a model on independent data because the original training set is locked away, how can you be sure how well it really works? It makes reliability checks very difficult.
So even if a paper reports, say, 98% accuracy, a hospital lab director needs to ask, okay, but will it work reliably on my patients with my equipment?
That's precisely the question. And it leads directly to the next big hurdle, the validation gap. Often, the accuracy metrics reported in research papers, they aren't directly tied to actual clinical requirements. For instance, what level of false positives for blast cells is considered acceptable according to hematology guidelines before it triggers unnecessary alarms. Many studies just don't benchmark against those critical clinical standards.
So the validation isn't clinically relevant enough yet?
In many cases, no. And rigorous external validation, testing the final AI model on completely new data from different labs, data it has never seen during training, that step is often missing or insufficient in the published research. As one source pointed out, even the public datasets can sometimes be too clean. They might have been pre-processed or had cells already perfectly segmented, which doesn't reflect the messy, variable reality of day-to-day clinical samples.
Okay, data and validation are huge. What about the types of things the AI is actually trying to find? Are there still gaps in what it can reliably do?
Yes, definitely gaps in coverage. While they're getting good with common cell types and some major diseases like AOL or malaria, the AI models still generally struggle with reliably identifying less common cell types. Also, non-malarial parasites, and particularly complex or subtle diagnoses, things like certain types of lymphoma cells that might appear in the blood or the really nuanced features seen in myelodysplastic syndrome.
And why is that? Just not enough data again?
Largely, yes. It comes back to not having enough high-quality, expert-labeled examples of these rarer findings to train the AI effectively. If it hasn't seen it enough, it can't learn it properly.
Makes sense. And finally, what about the people involved, the lab staff using this? Any challenges there?
That's a critical piece, the human factor. The sources definitely raise concerns about potential issues like over-reliance or confirmation bias.
How so?
Well, imagine an AI system flags a cell as potentially abnormal, maybe even suggests a diagnosis up front. There's a risk that busy lab staff might be tempted to just accept the AI's finding without performing their own thorough, critical review.
Especially if it's something rare that they don't see often themselves.
Exactly. Or conversely, if the AI misses something rare because of its data limitations, confirmation bias might lead the human reviewer to overlook it too, assuming the AI would have caught it. So there's a real potential for errors if the human expertise isn't used to actively validate and sometimes override the AI's output. It needs to be a collaborative tool, not a replacement you blindly trust.
Okay, so let's try and pull this all together now. We started this deep dive by looking at why analyzing these peripheral blood films is so important, but also why the manual process is slow and challenging. Then we saw the incredible progress AI, particularly deep learning, has made, generating really high accuracy numbers in research studies for identifying various blood cells, detecting diseases like malaria, certain leukemias.
Yeah, the technical achievements are genuinely impressive.
But, and it's a significant but, the sources make it really clear that despite this potential, we're not quite there yet for widespread, routine clinical use.
No, not as a full replacement. Certainly, the hurdles are substantial.
Revolving primarily around data needing more diverse, standardized, multi-center data.
Absolutely.
Around validation needing much more rigorous testing against real-world clinical needs and standards.
Crucial.
And acknowledging the current gaps in AI's ability to reliably handle rarer or more complex findings, often due to those data limitations.
Right. The overall consensus seems to be that AI has huge potential, but probably more as a pre-diagnostic tool or a powerful decision support system. Something to help the experts handle the more routine parts of the analysis faster, maybe screen slides and flag areas that need closer human attention.
So augmenting the expert, not replacing them.
Exactly. It's about making the human expert more efficient and maybe even more effective. And there is that really exciting, maybe further off prospect that AI might eventually be able to extract totally new kinds of information from these blood images. Higher dimensional data, subtle patterns that human eyes just can't perceive, potentially leading to new diagnostic insights down the line.
So wrapping up, this technology is clearly showing incredible promise in the lab, in research. It's automating tasks that take up a lot of time and skilled human effort right now. But getting it from the research paper into your local hospital's routine workflow, that means seriously tackling these fundamental issues. Data quality, data diversity, standardization, and really rigorous, clinically meaningful validation.
It really underscores a key point about bringing AI into critical fields like healthcare. It's not just about coding a clever algorithm. It's fundamentally about the quality, the breadth, and the representativeness of the data it learns from, and then proving beyond doubt that it performs safely and effectively against the strictest clinical standards before we can truly trust it with patient care.
Which brings us nicely to our final provocative thought for you, the listener, to mull over after this deep dive. Given this immense potential for AI to speed things up, maybe even find patterns humans miss, how do we make sure this incredibly powerful technology evolves carefully? How do we ensure it genuinely enhances that vital human element, the expert judgment, the clinical context, the compassionate care, rather than accidentally undermining it through things like over-reliance or data biases or gaps in validation? Something to think about.